For Lecture 10 for Bio 412 Comparative Genomics, we're going to be talking about epigenetics. And hopefully by the end of this lecture, you'll have a clear understanding of epigenetics, understand some of the mechanisms that underlie epigenetic effects, understand some of the methods that we use to study epigenetics, and also understand how epigenetics can change our understanding of concepts like biological concepts of things like heritability. So epigenetics. What is epigenetics, right? So it's different than just regular genetics, straight off the bat. When we talk about epigenetics, we're actually talking about our changes in the phenotype without changes in the genotype. Epi meaning on top of, so we're thinking about the stuff that's operating on top of our genetics. And what does this actually mean in terms of, biological entities? Well, we talk about things like changes in chromatin, i.e. like the shape or the availability of different parts of the genome, as well as other factors that can stabilize or destabilize gene expression programs. Often you'll see a term referred as the epigenome, right? So instead of epigenetics, we could also be talking about epigenomics. And similar to transcriptomics and proteomics, when we talk about the epigenome, this would be a global record of the regulator elements and chemical changes to the DNA and histone proteins of an organism, involved in regulating things like gene expression, development, tissue differentiation, and also the suppression of all different types of transposable elements. So again, these are the things that are operating on top of the DNA. Regulatory elements, chemical changes, affecting things like histone proteins that all are involved in the regulation and expression of different genetic elements. A couple terms that can be helpful when we're thinking about epigenetics. Chromatin, which is the term we use again to talk about where we might say we have gene-active chromatin or open chromatin. Heterochromatin, where we might say that this is generally an area of gene-repressed chromatin. These areas are often referred to as the light and dark areas of chromatin. We were going to talk about transcription factors, and again, these are DNA binding proteins that control transcriptional rates either by promoting or blocking the recruitment of RNA polymerase. We have different regions along the genome which are important to be familiar with. Promoter regions, which is a region of DNA that initiates transcription of a particular gene. Generally, these are located near the transcription start sites of genes on the same strand upstream of the gene. An enhancer is a short region of DNA that binds transcription factors, increasing the likelihood that transcription of a particular gene will occur. So also it's another element involved in gene expression and transcription. Typically these are cis-acting, so they're along the same strand of DNA or along the same chromosome, but they can be located pretty far away from the gene, up to maybe a megabase away, a million base pairs away. And they can either be upstream or downstream of where the transcription start site is. There are also insulators. These are genetic boundaries that block interactions between different genetic elements. And if we look at a diagram of how these factors might interact with each other, right, we have a gene over here in this area. And if we think of how sort of transcription functions, we have activator proteins that are going to be binding in these elements, these activator proteins attached to things like enhancers, and then they're going to help recruit a series of other transcription factors in addition to these activation proteins. And then all of these things will bind up here to this promoter region, right? And you see it sort of forms this big loop here where this enhancer and this promoter come together. as part of this process in the formation of this complex, we can then have the recruitment of RNA polymerase and then the transcription of the gene. Notice that we can also have some other groups out here that can be important. So for instance, there's an insulator site here where we might have binding of something like a CTCF transcription factor. And so this CTCF, if it comes and binds in, it can actually inhibit the ability or the type of gene which is actually expressed. And notice that there are some little methyl groups here that are blocking CTCF from binding to this region, right? And changing the conformation of this DNA and thus changing the ability of this enhancer and this promoter to come together and this gene to be expressed. So this is kind of your classic example of the different components that can be involved in transcription. And a couple things to note here. One, we have the shape of this process can really matter to this process in terms of what gets expressed. Notice that it's not just RNA polymerase coming in on its own. It depends on a whole suite of different things like promoters and enhancers, different transcription factors, different chemical factors like methyl groups or histone modifications that can be involved in each of these different components, all of them contributing to whether or not this gene is actually expressed. So it's not just one thing, it's a whole set of complex interactions with a bunch of different things occurring on top of this DNA that can help either promote or inhibit this reaction. When we talk about how we investigate epigenetics, basically we've got about 5 different ways that I'm going to be talking about today. We're going to be talking about DNA methylation, the role of non-coding RNAs, nucleosome placement, histone modification, and chromosome conformation. I think the classic example when we talk about epigenetics is thinking about the case of genomic imprinting. So when we talk about genomic imprinting, what we're talking about is that there's some sort of chromatin state that's defined by whether a gene or genetic locus is inherited from either the paternal or the maternal germline, so from the male and female germline. And what we understand this to be is that something about the gene is and its activity is affected based upon the parent of origin that chromosome was inherited from. One of the classic ways of looking at this is that we can see variation in the methylation patterns between whether a gene is coming in from a paternal source or a maternal source. X chromosome inactivation is one of the areas where this kind of process is thought to be important. So if we look at what goes on in this process, right, we have a partially pre-inactivated XP, so X chromosome coming from the father, right? It comes through and is progressively silenced early on in development, right? So the XM here is more active and is just being transcribed. So the maternal X chromosome is actually going to be the one which is going to be expressed in early development. Round about blastocyst site, we see a reactivation of the XP chromosome and then sort of random inactivation after that. And so after this stage, what we tend to see is that from the embryonic stage over, X gene expression is thought to be somewhat random throughout the organism. This is not always true. There are some specific instances in which we might see one X chromosome preferentially expressed over another one, but it's generally the pattern. So you see there's this distinct patterns of this expression at different points. At some point, we might see that there is going to be an X reactivation. There's also some imprinting erasure that happens at certain points in resetting of the X chromosome, right, where they and then sort of as they are passed into germ cells, we see an imprinting again of the XP chromosome when it goes in the placenta. So we have these specific patterns of X methylation that can be driven, and we see different patterns associated with these different X chromosomes and whether or not they're inherited from the mother or the father. An example of this is I used to work in a lab at University of Connecticut where they studied X-linked X-imprinted genes, and we would study them in mice. And for instance, there would be genes where depending, it was a locus called XLR, and depending on whether or not the mice we were looking at inherited an X paternal chromosome or an X maternal chromosome, they would have different gene expression patterns. And this was not due to any sort of difference in genetics. It was just whether or not they had inherited this this XP or this XM chromosome. And we used to actually test this in mice that we called X monosomics, so they only had one X chromosome. So we could see really distinct differences where we would have to worry about them being some sort of mosaic of X expression. Where we observe just one X chromosome being used in mammalian systems, where we have this X chromosome being inactivated, shows how you can have these parent of origin effects, right? So depending upon what's going on in the tissue, and if you're using, or that cell, and you're using the female or male inherited X chromosome, you can see different patterns of gene expression. The classic example of genomic imprinting is the IGF2 locus. And so in the IGF2 locus in mammals, what we see is that there's this difference in methylation patterns depending upon whether or not you're looking at the chromosome that's inherited from the mother versus the father, right? And so what happens is on the maternal allele, we have a region just past IGF2 where there is there could be methylation, but there is none. And when there is not methylation, an insulator binds in this site in between IGF2 and this enhancer, and it inhibits IGF2 from being expressed, and actually expresses a gene called H19 instead. Whereas the paternal allele has a bunch of methyl groups here, so CTCF cannot bind to this site, to the insulator site. Thus, the enhancer is able to actually bind to the promoter region and allow for the expression of IGF2. So in this case, depending upon if you're looking at the genome, the copy of the gene inherited from the mother or the father, you will see different patterns of gene expression. Exact same genotype, right? The only difference is the maternal genotype is going to be demethylated, and the paternal will be methylated. Why would things like this matter? Well, insulin growth factor can be involved in the growth of the embryo and the recruiting of resources, and it's thought that this is an example of concepts like sexual conflict, where the paternal allele is trying to recruit as many resources as it can for that specific embryo, whereas the maternal genome is trying to spread resources evenly across all of her possible offspring. So we have these two competing tensions, right, where one wants to have more gene expression, the other one wants to limit gene expression, and we could have these different possible outcomes because of this, and this may be what's driving this type of process. It's also thought that these types of factors may also be influenced by some environmental factors. For instance, there was a study done a while back looking at um, uh, uh, children born to some nomadic peoples in Africa, right? And their diet is very much seasonal and dependent upon the environment. And they noticed that there were a lot of what they called metastable alleles, which is a terrible term, but is accurate, where basically you have these alleles which can change, which are based upon these methylation patterns, which whether or not they're methylated or not is kind of context dependent on what the mother's diet is like. So we can see, and then that methylation state is then inherited and passed on to the offspring and affects their gene expression. So we can see how these kind of epigenetic effects could have some transgenerational components, and that's the thing that we're really interested in, because a lot of times in nature we see that there is an effect, an interaction between what the genetics is of an organism and what environment it's in. But we don't always understand how that interaction interplays. Well, epigenetic effects are an easy way for us to sort of explain that process as mechanistically. So how do we look at epigenetics? How do we assess it? Well, we've already been talking about one of the methods, and that's to look at DNA methylation. So when we're talking about DNA methylation basically means, in most cases, we're looking at cytosine, and sometimes in cytosine we'll find a specific region where there's actually a methyl group, a 5-methyl cytosine, instead of just a regular cytosine. And DNA methylation is generally believed to be the broadest controller of gene expression. It's about 70%. It fluctuates, but about 70% of the human genome on average is methylated. And specifically, one of the things that we look for are regions in which we find CPG sites. So parts of the genome where we have cytosine followed by a guanine, so it's a cytosine phosphate guanine site. And what we also tend to see is that these CPG sites, right, where we have CG, is not randomly distributed across the genome. So we find that there are regions that have high GC content, often referred to as CPG islands, and they have various levels of methylation, right? So these CPG islands are sort of ripe areas that we think can be associated with promoter regions and regions of variable methylation status. So there are actually different types of these types of CPG islands or regions that we think are important for important methylation sites. In addition to just CG, we could look at CHG or CHH, and H basically stands for, it could either be a C, or a T. So in other words, it could be a CAG or a CCG or a CTG. These are also regions where we might find some evidence of variable methylation. And you can see that the percentage of CPG methylation that occurs sort of through development, sort of your classic mammalian or vertebrate development, it changes, right? So we can start at about 70% in zygote stage, and then we can go down to as low as 25% in blastocyst stage, and back up until we get to somatic tissues, which are closer to 90% methylated. And then, of course, if you go down to like primordial germ cells, it gets very low. Everything gets demethylated, and then we see it sort of ramp back up into sperm, and less so in eggs, right? So it very depends. This methylation status changes, and one of the reasons why that is likely the case, right, is that different genes need to be active in different tissues or different parts of development. So how do we examine these patterns of cytosine methylation? Well, One of the classic ways of doing this is by doing something called bisulfite conversion. And so in bisulfite conversion, we basically take DNA and we treat it with we do a bisulfite treatment. And by exposing the sequence to bisulfite, basically we're taking regions where regions that are methylated are not really going to be affected, but regions that are CGs, right, where or any region that has a C, it's going to be where it's not methylated, right? We're going to see that cytosine converted into a uracil, right? So we can then test and see, like, when we go through and sequence these regions, we can then figure out which regions were methylated and which regions weren't, based upon the patterns of uracil substitutions. There's a bunch of different methods that we could do this. There's methyl-C-seq, there's BS-seq, there's reduced representation bisulfite sequencing, or RRBS. In the paper that we're going to be reading, they actually referred to BS-seq, I think is whole genome bisulfite sequencing, or WGBS. And they also do that synteny bisulfite sequencing, or SciBS, I think is what they call it. But basically, they're all of these methods are based off of the same sort of bisulfite conversion. They just take different approaches to figure out whether or not you're going to sequence everything or you're just going to sequence part of things, right? And so what they basically can do is the bisulfite sequencing is basically just taking the whole genome and treating it with bisulfite, and then sequencing everything and seeing what the patterns of uracil substitutions are. Reduced representation sequencing involves digesting your sample with an enzyme such as MSP1, which is going to be sensitive to methylation, right? And it's not going to cut at places where we think there could be where we have a CG and it's methylated. And so in this way, we're kind of able to target regions that are methylated versus regions that are not methylated. And because of that, we're able to not have to sequence the entire genome, but just sequence the component that we believe has a signal of methylation. So it can be a way of sort of doing a more targeted analysis. In addition to methylation, non-coding RNAs are another good way of epigenetic regulation. And when we think about non-coding RNAs, these are RNAs that are transcribed from DNA but are not translated into proteins. There are a wide range of these non-coding RNAs, and basically they can be involved in a variety of different things like processing of cellular products. And so we could be talking about things like small nuclear RNAs, which are involved in splicing, or SNO RNAs, small nucleolar RNAs, which can modify nucleotides in rRNAs and other RNAs. There is also a whole, I think the more sort of focused on targets of these non-coding RNAs are in their functions in regulating RNA expression and RNA translation. And in this way, we can see them acting to interfere with other RNAs. Specifically, we can see things like microRNAs, short interfering RNAs, PIWI RNAs, X inactivations, small linked RNAs, right? All of these different RNAs, even long non-coding RNAs, all of these RNAs mostly defined by either what they target or their size class. Many of these things, if you notice, that are very small, 20 to 30 something nucleotides long. And what they do is they actually target specific mRNA targets and bind to those targets, and then interfere with their ability to be translated into protein sequences. So there are a range of these different types of regulating RNAs, and so even though a gene might be heavily expressed, if there's a lot of microRNAs that are attacking it, then and binding to it, then that expression might be canceled out. If we think of kind of how these things work, it really sort of depends on which type of RNA family you're looking at. So again, there's a bunch of different types of these small RNAs. They can be transcribed in different methods. Historically, they're thought to form some secondary structures like these hairpin structures here that are then further processed down into the actual microRNA that they're going to form. And eventually we can see that what they're targeting for is either sort of, some sort of translational repression or promotion of mRNA degradation, or in the case of pewees, like targeting specific factors like transposons and their expression. So there's a whole set of machinery that's often associated with this process. So it's a fairly complex and involved process. Here's an example of how these mRNA expressions can be inherited epigenetically. So in this case, right, we have basically an example of an mRNA which is created and expressed off of the maternal sequence, and because there's a difference in the methylation status here, in this lower case, right, we see on the paternal allele this RTL1 mRNA is produced, and there's no mRNAs that are also produced, right? And so there's kind of directional transcription from in this direction, which allows for the transcription of the mRNA product and its translation. Meanwhile, on the maternal chromosome, there's no methylation, and so instead of this negative strand transcription, we actually get a positive transcription signal. And what this leads is to a whole bunch of hairpins being formed, being processed into microRNAs, and then these microRNAs, which are basically the complement for this mRNA, will then target that mRNA and cause that product to be degraded. So we can see like the two copies of this genome sort of in conflict with each other. Another classic example is that of pewees, or pewee interacting piRNAs, pewee interacting RNAs. These are very small RNAs, 26 to 31 nucleotides. They're widely found in everything from ciliates to mammals, but they've been heavily studied in Drosophila melanogaster. And their critical role is in the suppression of transposons, and they have this kind of ping-pong like cycle where you have sort of this sense and antisense primary transcript. It's almost like a game of Pong, bouncing it back and forth, right? Where you've got this antisense primary transcript. It's then cleaved. That this sort of cleavage process and prep process then creates something that can bind to this sense primary transcript, which then leads to cleavage, which then produces this product, which then binds to this antisense primary transcript, which then leads to cleavage, and you get the picture, right? So what happens in this process is you have this interaction of this ping pong cycle with this OB gene, and this AGO3 gene, and this is thought to be largely an epigenetic inherited process because typically peewees are passed on maternal lineages because they're actually inherited through the egg and not through sperm. So to sequence these microRNAs, a lot of times what we'll do is we'll take these short RNAs and we'll ligate on adapters and then use those adapters to get a real-time primer onto and form a sort of double-stranded end, add some other adapters on, and then allow us to sort of make a reverse transcribed product. Then once we have that sort of cDNA product, we'll then use PCR to amplify that target, and then we'll make sure we do some size selection to make sure that we're only sequencing things of a very specific size range. And then this allows us to sequence this thing, and basically what happens is that microRNA is going to sit in between some indexes that we can already read and understand. It's kind of how we target these small RNAs, right? So we just basically ligate some adapters onto it, make it into cDNA, and sequence it. Another method of epigenetic control is examining nucleosome placement. When we think about nucleosomes, what do we think about? These are octamers, right? They are generally 2 histone H3, H4 dimers and 2 histone H2A and H2B dimers. There are various histones that can be replaced. There are other variants that can replace these if we need to. But its main function, right, is that these nucleosomes placements allow for the folding and packing of DNA, and that physical process and the chemical structure of and the status of these histones can block or promote RNA polymerase in specific regions. And so this can heavily affect the shape of DNA and therefore perhaps the accessibility and openness of that DNA to be transcribed, as well as specific post-translational modifications that can help promote different types of activities. So how do we examine things like nucleosome occupancy? One of the ways is to do things like what's known as MNA-seq. And this is where we would isolate chromatin, and then basically we do something that's called cross-linking, where we treat a sample with something like formaldehyde or paraformaldehyde, which binds these histone proteins to the DNA, right? And so it forms this very strong covalent bond, so very stable. And that's what pretty much happens, right, is that how these histones work is that DNA has wrapped itself around the histone, right? And so what happens is after we've cross-linked everything, right, and DNA is wrapped around it, we'll then go through and do an eminase digest. And what the eminase digest does is it digests everywhere that doesn't have isn't bound to a nucleosome. And so what it allows you to do is isolate the parts of the genome that had nucleosomes attached to them, right? After you've done that, then you can use an antibody to pull down this cross-linked chromatin and then unlink the DNA and the protein so now you can separate them, and then we can sequence this DNA, right? So we can sequence this resulting chromatin that was previously bound to these histones. And this is an example of sort of like the kind of result that we might get, because after we're done with that sequencing, we'll then take those sequences, we'll map them back to our genome, and we can compare, what are the patterns that we see. And what we can do is we can see, like, say we have 2 different samples, we can say which ones do we have examples of mapping to specific sites and which ones don't we. And so we might think that, okay, nucleosome varied in this case because the red sample didn't have nucleosomes bound in either of these two regions. So in addition to nucleosome placement, we can actually also look at how epigenetic control is conferred through the modification of the histones that make up these nucleosomes. And just a reminder, when we're talking about these histone modifications, what we're really talking about is the process of post-translational modification, right? So where we go from genome to transcriptome to translation, and then before we actually get the full activation, right, there's often a bunch of post-translational modifications that proteins can undergo before they hit their full activity. And these post-translational modifications, it's usually an enzymatic process. There's a whole bunch of different types of post-translational modifications. There's glycosylation, phosphorylation, acetylation, oxidation, methylation, ubiquitination, etc. Right? So there's a bunch of different things. And notice that specific amino acids are prone to specific types of post-translational modifications. So not all of them will have the same ones. A lot of these patterns of post-translational modification are pretty organismal dependent and tissue or location dependent. And they're also dynamic. They can be stable or dynamic. So sometimes they don't change ever, and other times they're moving around and changing all the time. What we do know, though, is that these post-translational modifications to proteins will have an effect on biological activity and can be pretty important. And so here's just an example of where you have a lysine at the 56th position of the H3 histone, right? And in one case, that site is acetylated, as evidenced here by this little sort of blue piece, and otherwise in other up here it is not. And what we tend to notice is that there are going to be some slight conformational changes and other changes to these molecules depending upon whether or not there is an acetylation of the specific site. When we talk about epigenetics, there are specific histone modifications that are important. And so again, when we talk about histone modifications, what we're talking about in these post-translational modifications, covalent ones, two histone properties that carry specific information that largely will tell the cell and other pieces or indicate whether or not a specific region of the chromosome is either euchromatin or open chromatin or heterochromatin, dark chromatin. And so you can see this is sort of an image of each of these 8 histones sort of stacked up into nucleosomes, right? And each one of these histones, we think about things like the H3 histone, it'll have sort of different types of arms coming off it. And for each of these arms, we can identify specific post-translational modifications that can have very specific relationships or be great indicators of specific chromatin states or functions of different areas. Largely, we're looking at the H3 chromosome in this H3 histone in this case, and one of the things that you'll notice is that these are all the possible modifications. A lot of them are happening to lysines, which is indicated by this K there. So methylation, phosphorylation, acetylation, ubiquitination, sumolation, and how we identify these sites. So say it was acetylation at the K27 position, right? So we would call this H3 K27 methylation if we're going to talk about this specific modification, or H3K9 acetylation in this case, in this position. So that's how we can identify which spot we're looking at. Again, what happens is we could have our closed chromatin period, right? And then if we suddenly these H3 or other components become acetylated, they'll actually be disentangled from the chromatin and allow it to open up and allow all the transcription factors and polymerase 2 to all be sort of recruited into this area. When we think about epigenetics, there's a whole suite of different types of functions and mechanisms we could associate with epigenetics, but a lot of it is involved in sort of this understanding of histone modifications and how they can be informative on understanding these types of processes. So there are a bunch of different types of these modifications. Over here in this chart, I think this is kind of an important chart because these are some of the standard ones that are used in the field. So H3K4ME3, which is methylation, a 3-methylation of the fourth lysine in the H3 histone. Would indicate an active promoter, and H3K4ME1 modification would indicate an active enhancer. H3K27 acetylation shows both promoter and enhancer activity. H3K27ME3 can show an inactive chromatin. RNA polymerase can show where transcription is happening. H3K4ME2 can show where euchromatin is, and H3K9M2 can show where heterochromatin is. And so these are all sort of some of the major modifications that we might want to be aware of. In addition to this, we can see that there are like hypotheses about how this code kind of works. So one of them is the histone code hypothesis that says, writers produce the combinatorial patterns of histone modifications that specify distinct biological outcomes by recruiting effector proteins or readers. And one of the thing about these modifications, right, is they don't work in a vacuum, so they interact with DNA methylation and small RNAs to bring about various different chromatin states. And another thing to think about is that these systems are pretty dynamic. So nucleosomes can move around throughout the genome, so as long as they're locked into place. And almost all of these marks are reversible if we wanted to reverse them. So an example of where these types of processes can be informative can be things like looking at the status of induced pluripotent stem cells. So in this case, blocking specific histone modifications can actually lead to the better formation of induced pluripotent stem cells. So there are some specific ways that we can use things that happen in the epigenetic space to manipulate systemic outcomes and change the biology of systems. How do we assess these types of processes? Well, the classic way to look at something like histone modifications is something called a ChIP-seq, which ChIP stands for chromatin immunoprecipitation sequence. So in this case, it's kind of similar to the MNase example, except that you're going to cross-link your DNA with your proteins that are bound to it, and then you're going to chop everything up, and then what you need to do is your immunoprecipitation component. And so what you do is you use a specific antibody that targets like a histone that has been acetylated at position 27, right? And so by using that process, you can then pull down the specific DNA complexes that are associated with the modification that you're interested in, and then uncross-link or break those apart, and then you can isolate that DNA and sequence it. And then once you sequence it, then you can map it back to your genome, and you can show like the different patterns of where these reads bound, which show where on the genome these different post-translationally modified histones were. So we can see, like from a comparative standpoint, you can do things like compare the status of different histone modification patterns across species. You can look and say, okay, where these things were bound. Do we see any specific motifs that are shared between things like chicken and lamprey and human that help us understand how the things like epigenetic control have evolved? And even you can compare sort of the placements of these processes to better understand how gene expression has evolved over time. And in this case, like by looking at, say, lamprey, chicken, and human, we can find examples of where like CTCF binding became particularly important in things like Hox gene expression and regulation. Lastly, there is the last sort of way of doing that we think of epigenetic control is by looking at things like chromosome conformation. So in chromosome conformation, remember we had that little picture of gene expression, we had that loop, right? In this case, we have the recognition that there is in fact three-dimensional structure in biological systems, and sometimes that matters because it can take two things that are really far apart and bring them close together. And that's important for a wide range of reasons. We can also make these big loops, and if I have something here that's being expressed and I have something next to it that's being expressed, it may be that RNA polymerase is going to become entrained in this loop and then preferentially sequence what's here versus maybe preferentially not sequencing what's over here, right? So there are some ways that conformation can really matter in understanding about what's influencing the expression and the activity of different genetic elements. So there's a bunch of different ways to do these chromosome conformation assays. Classic way is to do something like 3C, which you can basically just use qPCR to do. Then there was 4C, which was more sort of focused towards microarrays and sequencing. And then a whole bunch of other newer approaches. The most popular of these being high C, where basically what you're going to do is you're going to take your chromosome, you're going to cross-link it, and then you're going to fragment it and break it up so that it forms these complex where you have your nucleosome bound DNA in the center. You then do a ligation step, and instead of adding in sort of primers that are going to help each end ligate, in this case you're trying to get the ends to ligate to each other. So what happens is you end up with a piece of DNA after you reverse cross-link where you have a blue section and a red section. And if this indicates here, right, where blue is on top and red is on the bottom, it could be that you have, two pieces of DNA that you can sequence together that are, approximately, I don't know, it could be 100,000, a million base pairs apart. And by looking at this type of data, what you can find is, like, if you do things like look at Arabidopsis and compare endosperm to leaf tissues, you can see that there are very different patterns of these in chromosome conformation, depending upon if you're looking at different types of tissue. And in this case, what you end up having is you have these systems where you have like more of these marks where they have like lots of interactions, or they have sort of more complex examples where they have fewer interactions with other entities, but lots of interactions among themselves, right? So the way that the chromosome becomes either more accessible or less accessible to transcriptional machinery based upon these types of conformations. And over here you can kind of see, like, you have these structural domains which have like sort of a lot of high C data like compressed into those regions, versus these loose structural domains which kind of allow for more open chromatin, more access, perhaps more expression. And so we can see, sometimes the patterns are pretty similar between the leaf and the endosperm on these chromosomes, and other times they can be very different, right? So, and you can kind of see how this looks like, endosperm in this case ends up being a lot more structured versus leaf, which tends to be a little more open. And so in this case, like the blue components, which are more open, are generally associated with like the arms of the chromosome, whereas these compressed areas are sort of more associated with heavily repetitive elements and things like that. All right, so that's an example of, that's a discussion of the different ways that we might think about epigenetics and some of the different tools that we use to examine it, all those different mechanisms. And again, the idea is just that epigenetics is things that happens on top of the genome, on top of genetics. So they show us patterns that we wouldn't be able to detect just by looking at the DNA sequence. And that these factors can actually have a pretty significant effect on our biology.